Hello students, this is experiment number 9, urine test for presence of sugar and albumin. This experiment is divided into two parts, 9.1 and 9.2. Let us start with experiment 9.1. Its objective is to detect the presence of sugar, that is glucose, in urine. Materials required for the experiment are: we need urine sample, dropper, test tube stand, test tube holder, spirit lamp, and we need Benedict solution, Fehling solution A and Fehling solution B. Theory of the experiment. As you know, so many metabolic reactions takes place in our body and a large number of waste products are formed in our body during these various metabolic reactions. And out of all waste products, urea is the principal nitrogenous waste product which is excreted in the form of urine along with other waste products. Urine is yellow in color and it contains 95% to 96% water, 2% urea and 2% other dissolved substances. But persons suffering from some metabolic disorders may excrete glucose or albumin or acetone or bile salts in their urine. And if sugar is detected in urine, it means person is suffering from diabetes mellitus. Come to the procedure used to detect glucose in urine sample. First test is Benedict's test. For this, first take 2 ml of urine sample in a test tube and add 2 ml of Benedict's solution in it. Now, boil this mixture and then cool it to room temperature. You may notice the green, yellow or brick red precipitates in the mixture. See the second test, it is the Fehling's test. Here also take 2 ml of urine sample in a test tube and then add 2 ml of each of Fehling solution A and Fehling solution B. Then boil it for about 2 minutes and cool it to the room temperature. We can see greenish yellow, yellowish red or reddish orange precipitates. This is the diagram showing urine test for glucose. Draw it in your practical file. Write your observations of both the tests in this observation table. Then write the results. As per the observations, given sample of urine contains glucose. Now write the conclusion. In Benedict's test, green precipitate shows traces of glucose, yellow precipitate shows moderate quantity of glucose, while brick red precipitate shows intense quantity of glucose in urine sample. In Fehling's test also, greenish yellow shows traces, yellow red shows moderate and reddish orange precipitates indicates large quantity of sugar in the urine sample. Finally, write the precautions to be taken. First, wash the test tubes thoroughly before use, and second, use standard reagents for doing the experiment. This is experiment number 9.2. Its objective is to detect the presence of albumin in urine. Write requirements. We require urine sample, test tube stand, spirit lamp, sulfur salicylic acid, Roberts solution and dropper. Write the procedure. First test to detect the presence of albumin in urine sample is sulfur salicylic acid test. Here, first take 2 ml of urine sample in a test tube and add an equal volume of 30% of sulfur salicylic acid in it. Now, heat the tube gently. We can see a whitish or cloudy turbidity in the solution. You can see these in the picture given. Second test is Heller's test. Take 5 ml of Roberts solution in a test tube. Now, tilt the tube and add urine with the help of dropper on the inner side of test tube so that it forms a layer of Roberts solution. This step you can see in the first picture. Now see the second middle picture where a white ring is formed at the junction of Roberts solution and urine sample. These diagrams you have to draw in your practical file. Write your observations of the test done in this observation table. Write results. As per observations, urine sample contains albumin. Now write conclusion. In sulfur-salicylic acid test, the whitish or cloudy turbidity shows the presence of albumin in the urine sample. And in Heller's test, formation of a white ring is an indicative of the presence of albumin in the urine sample. Finally, write these two precautions to be taken while doing the experiment. So, that's all for today. Thank you very much for staying with me. Subscribe the channel to get the information about my new video. Thank you so much.